This conference will now be recorded. This conference will now be recorded.
Hello students, I'm Dr. Divya from TeenEinstein. So now we are in the 6th chapter, molecular basis of inheritance. So in the previous classes, we have learned about all this DNA structure, then different types of RNA. Then we learned about this semi-conservative model, DNA double helix. Then the difference between this repetitive DNA and mRNA, tRNA, then what is template strand, what is coding strand. So now we have learned about so many terms of this molecular level biology, right? So in today's class, we are going to learn about Human Genome Project. So this is about the application. We are going to learn about application or where we can apply this molecular level studies of gene and DNA, or what is the use of this molecular level studies of or molecular biology, where we can apply this. So this inheritance studies and all we can apply in different levels. Okay, so these are very important topic, especially for NEET exam. You may get so many questions. So first, we are going to start with Human Genome Project. Okay, this Human Genome Project question is very important for your NEET exam and for your board exam, because Human Genome Project, you may get a question like, what is the importance of this Human Genome Project, or why it is called as a mega project or something for your board exam. And from this, the size of this human genome or such type of MCQ questions are also made. So it's important for both exams. The human genome means, generally we can say that haploid set of chromosome. When we are considering about haploid set of chromosome, what do you mean by human genome? Or you know that genetic informations, the genetic informations of an organism, that means the base sequences present in the DNA of an organism, right? Or the DNA sequence that determines the genetic makeup of an organism. Or haploid, the information is present in haploid set of chromosome of human gene or human genome. Okay, so the DNA sequences which determines the genetic makeup of an organism. So if two individuals are different, that means their genetic or their DNA sequences also shows some kind of differences. If that whole sequence that is not different, means at least in some places that shows some kind of differences. Okay, the whole sequence may not be different, but at least in some places that shows some kind of differences. So these kind of informations are this kind of minute differences that gave motivation to scientists, and they found out the complete sequence of human genome. Can you imagine the complete sequence of human genome? So you know that it's not a simple process. Okay, So you know now genetic engineering, it is a fast growing field. There are so many findings. It's not there for 11th and 12th standard, but in future, if you are going in biology field, you can learn about all these things. It's a fast growing field. So by using these techniques, what and all we can do? It's very easy now to isolate and clone DNA or any part of DNA, right? And then by these techniques, it's very easy to find out the DNA sequences. So all these led to the launching of Human Genome Project in the year of 1990. So they launched the Human Genome Project. They started that in the year of 1990. Okay, so Human Genome Project, it was a mega project because human genome is having approximately 3 into 10 raised to 9 base pairs. Got it? So it's a mega project and human genome is having around 3 into 10 raised to 9 base pairs. Okay, it's a large number. So the genetic engineering, it's very fast growing field, and with the help of these brands, we can easily isolate DNA and we can clone DNA or any part of DNA we can clone now, right? So it's a very It's a very useful technique. So we can find out the DNA sequences. So all these fast growing fields, that with the help of those techniques, they launched the sequencing of human genome. And it was launched in the year of 1990. So Human Genome Project was called as a mega project. Human genome, it is having around 3 into 10 raised to 9 base pairs. Got it? So with the establishment of genetic engineering techniques, where it was possible to isolate and clone any piece of DNA. Okay, it's very easy with all these techniques. They made it easy to isolate the isolation of DNA and cloning of DNA. What do you mean by cloning? Creating exact piece of that, cloning of that DNA. And availability of simple and fast techniques for determining DNA sequences. So with the help of all these, they launched the project of human genome sequencing in the year of 1990, and Human Genome Project was called as a mega project. Human genome is said to have approximately 3 into 10 raised to 9 base pairs. Note down, 3 into 10 raised to 9 base pairs that is having human genome. Okay. And just now we said it was a mega project because human genome, it is almost 3 into 10 raised to 9 base pairs. So sequencing of this genome, it was too costly and it was time taking. It was too costly because you can imagine 3 into 10 raised to 9 base pairs means it's a large number. And sequencing of all these, the whole genome, definitely it will take so much time and it may be costly, right? So the cost of that was around $3, $3 per base pair. Okay, so like that, 3 into 10 raised to 9 base pairs are present. So per base pair, it was $3, right? So like that, how many should be sequenced? 3 into 10 raised to 9 base pairs. So it was around $9 billion. The cost was around $9 billion. Okay, so it was a costly project and time-consuming project. Why it is called as a mega project? Around 3 into 10 raised to 9 base pairs are present, and the cost of sequencing per base pair $3, and like that, the total cost was around $9 billion. So it's a huge number, right? So if it's getting sequenced, that sequences were to be stored in books, and in the type format they need to store this. So the number of sequences itself, that much, right? So how we can store this data in typed format? Then just imagine in book, each page will be having 1,000 letters, and like that, each book contains 1,000 pages. Each book will be having 1,000 pages, and each page will be having 1,000 base pairs, right? So like that, how many books required? Around 3,300 books. So for storing the information of DNA sequencing from a single human cell, okay, from a single human cell, for storing the data, we need 3,300 books, and in each book, 1,000 pages, and in each page, 1,000 letters. Okay, so huge amount of data is required. Huge amount of data requirement is there for the storage of this, and we need computational devices for the storage and then analysis of data. So due to all these, Human Genome Project, it is called as a mega project. First of all, 3 into 10 raised to 9 base pairs. Base pairs, so many base pairs, right? And the cost of this, it is a time consuming project and costly project. And the cost is around $3 per base pair. So like that, 3 into 10 raised to 9 base pairs. And the total cost was around $9 billion US dollars. And that storing, again, after sequencing, next comes the storage of data. Where we will store this data? This data, if you want to store the data, that need 3,300 books, and each book should have 1,000 pages, and each page should have 1,000 letters. So like that, it's a huge data. That's what it means. Okay. So because of all these, Human Genome Project was called as a mega project, Human Genome Project, or HGP. It is called as a mega project. So this Human Genome Project is also associated with the development of bioinformatics, and bioinformatics, it is a new research area of biology with information technology. Both are combined. So that area, that branch, it is called as bioinformatics. So that made the research more easier. Okay. So which made this molecular level analysis into simple methods. Got it? So the cost of sequencing required is US dollars, $3 per base pair. So the total estimated cost of the project would be approximately 9 billion US dollars. And if you obtain sequence, where to be stored? How this will be stored? So in typed format, in the form of books, each page of the book contains 1,000 letters, and each book contains 1,000 pages. Then 3,300 such books will be required to store the information of DNA sequence from a single human cell. Okay, that's the main thing. From single human cell, we need this much, or we are getting this much data. Okay, so the enormous amount of data also necessitated the use of high-speed computational devices for data storage and retrieval and analysis. For data storage, data retrieval, and data analysis, high-speed computational devices are required. And Human Genome Project was closely associated with the rapid development of a new area in the biology. New area in biology that is called as bioinformatics. Got it? So because of all these reasons, Human Genome Project was called as a mega project. Next, goals of Human Genome Project. So in what are all the goals of Human Genome Project? Approximately 20,000 to 25,000 genes. Identification of 20 to 25,000 genes in human DNA. Okay. This is another important question for board exam. Goals of Human Genome Project. First question, important question is why it is called as a mega project. And now the goals of Human Genome Project. Identification of 20,000 to 25,000 genes in human DNA. Then determine the sequence of the 3 billion chemical base pair that makes up human DNA. Okay, 3 into 10 raised to 9 base pairs are present, we said. So we need to determine the sequence, determination of the sequences of these base pairs, chemical base pairs, which is the base of human DNA. That is another point. And third point, this store this information in databases. Next comes after sequencing, again storage of this information in databases. Then improve tools for data analysis, for analysis of this data to find out. For research findings, we need to analyze the data, and for that, improved data analysis tools. Okay. Then transfer related technologies to another sector, other sectors such as industries. Then we need to bring all these informations into application level. Then address the ethical, legal, and social issues that may arise from the project. Okay, ethical, legal, and social issues may come. So for that, that also should be addressed. So what are the goals of HDP? Identify approximately 20 to 25,000 of genes in human DNA. Then determine the sequence of 3 billion chemical base pairs that makes up human DNA. Then after sequencing, storage of the information in databases. Then for analysis, improved tools, data analysis tools. Then transfer related technologies to other sector. Application of this technologies. Then addressing the legal, social, and ethical issues. Okay. So these are the goals of Human Genome Project. And next is this Human Genome Project. It was around 13 years. So it is a 13-year project, and that was coordinated by US Department of Energy and National Institute of Health. Okay. It was coordinated by US Department of Energy and the National Institute of Health. Okay. Then during the starting years of this Human Genome Project, Wellcome Trust, UK-based Wellcome Trust became the major partner. Okay, Wellcome Trust is a charitable foundation which is focused on the health research, and it is based in London, UK, and they are releasing fund. Mainly, they are giving fund for the improvement of human and animal health. Okay, that is Wellcome Trust is a charitable foundation, and it is based in UK, London, in UK, and they are mainly giving funding for the improvement of human and animal health. For such research works, they are giving fund. Okay, so during the early years or starting years, Wellcome Trust became a major partner in Human Genome Project, and additional contributions. It came from France, Germany, China, Japan, and all. They gave additional support, and that project, the mega project, a 13-year project that got completed in 2003. Okay, in 2003 that got completed almost. Then we know that variations in DNA sequences, and the idea about that, or the knowledge about that helped in the treatment and diagnosis and treatment of so many human disorders. Okay, so these Human Genome Project and DNA sequencing that helped, or that led to the understanding of human biology. The main or the important point is this Human Genome Project. What we were doing in that Human Genome Project? Identification of human genes, right? And their functions. So in such findings, from that, through this human genome project, the DNA sequencing also they did. So that led to the understanding of human biology, and that gave many clues about the diseases or the disorders which are present in, or up to that level, it was very, or up to that time it was very difficult, or the idea about such diseases were very less. So after human genome project, such things became very easy. So in the case of health issues or health diagnosis, that treatment and so many human disorders, this knowledge of human genome project was very helpful. So these human genome project and DNA sequencing led to the understanding of human biology, mainly in human biology, and that this kind of researchers or the analysis that gave clues about non-human organisms DNA sequencing also. So like this, we can sequence the DNA of organism. So in research field, they got an idea like this. To find out the function of each gene, we can go for sequencing of genome of organism. So they got such an idea in research field. So they were mainly helpful in the field of agriculture. In agriculture field, it was helpful. In human health, it was helpful. So like this, in so many fields, it became so helpful, this human genome project. And many human genome, many non-human genomes. Like this, many non-human genomes also sequenced. For example, in Drosophila, they sequence, in bacteria, yeast, etc. In all these kind of organisms also, the sequencing they did. Because they got already an idea about the sequencing, genome sequencing. So the same trial they did in other organisms also. And in the case of Drosophila, bacteria, yeast and all, in the same way they did the genome sequencing. Okay. So human genome project that or HGP, that has revealed that there are probably about 20,500 human genomes, no doubt 20,500 human genomes, and the ultimate product of this HGP or human genome project that has given the, in world they created something very big, right? They gave a good idea and they gave a resource of detailed information about what? About the structure, then organization and function of the complete set of human genes, right? About human genes, all genes they sequence. So they got an idea about the structure of that gene, function of that gene, and how it is working in human body. So they gave a complete idea after this human genome project. So that was the ultimate product. The ultimate product of this human genome project was that become a complete resource for the detailed information. All information they got about human genes, right? All genes, they gave complete idea about the structure, function and organization of complete set of human genes. So all these informations that can be considered as the basic set of instructions, right? For all development and function of human beings. We can make out which gene is working and which gene is working for what purpose. So that was the main finding or main goal, or that was the main output of this human genome project. So the tools created through this human genome project, that is also used to characterize the entire genome of several other organisms. Just now we said, like, from this idea, they got a chance to sequence other organism also, or the genome of other organisms also. They created entire genome of several other organisms, the sequencing of several other organisms also there, and these are mainly used in biological studies. In which studies? For studies of organisms like mice, fruit fly and all, they applied the same technology. So why they made all these attempts? Because all these studies, they support each other. And how it is getting support or how it is getting a new idea. See, most of the organisms, they have similar or homologous genes. Right? And almost they have similar functions also. Got it? So, if they are sequencing one genome with in other organisms also, such genes will be present which is having almost same function. So, it is very easy to identify the sequence or the function of a gene in the other organism also of these kind of model organism. That may help to find out the function and structure in other organisms also. So this kind of ideas that can be used in biomedical technology, then other research areas also this will be useful. So that is the importance of Human Genome Project. Okay. So the knowledge about effects of DNA variation among individuals can lead to the revolutionary new ways to diagnose, treat, and someday that's prevent thousands of disorders that affects human beings. Right? So through this, they were able to find out the structure and function of genes. So such findings that helped in the diagnosis and treatment of diseases, and mainly they were able to prevent the disorders that affect human beings. So besides providing clues to understanding human biology, learning about non-human organisms' DNA sequences can lead to an understanding of their natural capabilities. So this understanding of human biology that helped in the learning of non-human organism and their sequencing work also done. So that can be applied towards solving challenges in healthcare, agriculture, energy production, environmental remediation. So in all these fields, this can be applied. So these are all the plus points of Human Genome Project or the outputs of Human Genome Project. And many non-human model organisms such as bacteria, yeast and all, and Drosophila, all these also sequenced. They got sequenced. Okay, sequencing of all this also done. Got it? Next, mainly two methodologies. Two methodologies were used. One is expressed sequence tags or ESTs. Okay, expressed sequence tags. ESTs, short form is ESTs. Just read that, expressed sequence tag. The sequences which are expressed. That means the coding regions, the exons, the expressed sequences are getting tagged. That is one method. Okay. So in expressed sequence tag method or an EST method, how it was working? Identifying all the genes that are expressed as RNA. So in the first method, expressed sequence tag, how it was done? Identification of all the genes that are expressed as RNA, which are expressed as RNA, those genes are identified. That was one method. And then second method, the second method, it is a sequence annotation. Okay. Second method is sequence annotation. In this method, this is like actually just like a blind approach. Why we are saying like that? Because in sequence annotation method, in this sequencing, the whole set of genome is done. In EST or expressed sequence tags, only expressed RNAs, only that genes which are expressed as RNA, they are considered. But here, the whole set of genome that contains the coding and non-coding regions. The whole set of genome will be having coding regions and non-coding regions, right? Here you can see EST means here only exon 1, exon 2, exon 3, exon 4. Here only expressed sets are there. Only that one is getting tagged or that one is identified. Okay, but in the case of sequence annotation, in sequence annotation, coding regions are there, non-coding regions are there. The whole thing is getting sequenced. If it is coding or non-coding, everything is getting sequenced. So that's why we are saying it as a blind method. And later assigning different regions in the sequence with functions. First, everything is getting sequenced, then assigning the function for that coding regions. Okay. Here you can see in this diagram, you can see the analysis of coding and non-coding regions. So in second picture, in first you can see only the coding regions. Only expressed sequences are marked in first one. Exon 1, 2, 3, 4. All these in genomic DNA. So in EST here you can see only the coding regions are marked. Okay. But in the second one, non-coding analysis, non-coding and coding both. Everything is sequenced and after that assigning function for the particular region. Okay. So the two methods are involved, two major approaches. One approach focused on identifying all the genes which are expressed as RNA and it is called as express sequence tag. And the second method, it is a blind approach of simply sequencing the whole set of genome and that contain all the coding and non-coding sequences and later assigning different regions in the sequence with functions. Okay, this is called as sequence annotation. Then two methods, one is EST, express sequence tag, and second method is sequence annotation. Okay, and next, now we are going to learn about the procedure or the process of this methodology or how sequencing is done. So let's see. First step is DNA isolation. For sequencing, first we need to do, first we need a DNA, right? So from human, the DNA is getting isolated. That is the total isolation. Total DNA is getting isolated from the cell. From cell, total DNA is getting isolated. And next step is conversion into random fragments with smaller size. Then the whole DNA is getting isolated, then making that into random fragments, making that DNA into random fragments with smaller size. Why we are making into fragments of smaller size? You know that DNA is a very long polymer, right? DNA is very, very long polymer. So technically it is very difficult to sequence a long piece. A long piece of DNA, it is technically very difficult to sequence like that. So that's why we are making into random fragments with smaller size. Got it? So using that specialized vectors, these DNA fragments are cloned in suitable host. First making DNA isolation, then second step is making them into smaller fragments, random fragments, then cloning of these vectors, cloning of these DNA fragments with specific vectors with suitable host, and this cloning. After cloning, what will happen? This cloning will result in the amplification of each piece of DNA fragment. Cloning results in the amplification of this DNA fragment, each piece of DNA or this DNA fragment, and that makes the sequencing more easier. Okay, and commonly used hosts, which are the commonly used hosts? Bacteria and yeast. So the vectors are called as bacterial artificial chromosome. Bacteria and yeast, these are commonly used hosts. Okay, so the vectors are called as bacterial artificial chromosome or BAC and yeast artificial chromosome. Note down, vectors, commonly used vectors are bacterial artificial chromosome or BAC. and yeast artificial chromosome. Yak, back and yak. Okay. So for sequencing, first the total DNA is isolated from the cell and then it is made into random fragments or for small or with smaller size, because the long polymer of DNA is very difficult. Technically, it's difficult to sequence. So first we are making that into smaller sizes, then cloning in suitable host. How cloning is done? By using specialized vectors. And this cloning helps in the amplification of each piece of DNA fragment, and that makes the sequencing easy. Then the host, which are the commonly used hosts? Bacteria and yeast. And commonly used vectors, they are bacterial artificial chromosome and yeast artificial chromosome. And next, in next step, these fragments were sequenced. Okay, these fragments, now it is in fragmented form, so that fragments were sequenced using automated DNA sequencer. Okay, these fragments were sequenced with automated DNA sequencer, and this automated DNA sequencer that works on the principle of a method which is developed by Frederick Sanger. Sanger's method. There is a method called Sanger's method. So based on that, this automated DNA sequencer is working. Okay. So Sanger, he is also credited for developing methods for the determination of amino acid sequence in proteins. We have already discussed about Sanger, right? The scientist credited for the developing method. He credited for the development of method for amino acid sequencing. For amino acid sequencing, Sanger's method is there. Okay, so he is having already a credit of amino acid sequencing. Okay, based on that, on Sanger's method, this automated DNA sequencer is working. Okay. Then after sequencing, now automated DNA sequencer with the help of that sequencing over, then after sequencing, these sequences were arranged based on some overlapping regions. Okay, there will be some overlapping regions present on the sequences, and in this condition, that so in that condition, that require a generation or the development of overlapping fragments. Then only the sequencing will happen, right? The overlapping fragments should be formed. Then only the sequencing will become easier. So alignment of these sequences, alignment of these overlapping sequences, it's not easy. We cannot do that. Human power cannot do that. Okay, they can't fix the problem. So for that purpose, computer-based programs were developed. Okay, there comes the computer-based program, application of that, different softwares. Then annotation of all these sequences were done. Then these annotated sequences were assigned to particular chromosomes. Okay, so what are all the steps? First, isolation of DNA, then making into fragments, then with the help of vectors, cloning, then host, bacteria and yeast, they are the host. Then these fragments were sequenced with the help of automated DNA sequencers. Then these sequences, they are having overlapping regions, and arrangement of these overlapping fragments, it's not possible by human beings. So there comes the software applications. Then specialized computer-based programs were developed, and these sequences were annotated and they assigned into particular chromosome. Each chromosome, where it is matching, they are assigned to that particular chromosome. So these are the procedure. Then in the month of May, they completed 2006. In 2006, they completed chromosome number 1, and that was the last one of 24 chromosomes. 24 chromosomes, how we can say 24 means 22 autosomes and 2 sex chromosomes, X and Y. Okay. So the sequencing that got completed, the final one, chromosome 1 was completed in 2006 May, the last sequencing. And that was the last of 24 chromosomes. All those got sequenced. Okay. So next task, sequencing over. And next they need a genetic map, or the mapping of all these chromosomes should be done. Then only we can find out this gene is present on this chromosome and that is doing a particular function. Okay. Each gene that should be located on particular chromosome, that is called as genetic mapping. So what is genetic mapping? Genetic map means that is a type of chromosome map. In genetic map, it shows the probable or relative location. We can't exactly locate, almost in that location that gene will be present. Like that we should be able to say. So that is called as genetic mapping. And some other features of all these things. One gene is coming in between these genes. Like that, there are some kind of relative locating of genes. So in a map, you can see genes. See, by then, suppose here on this chromosome, here one gene is present and another gene is present here down. So the distance is more. Right? If one gene is located on top and very next to that another gene is located, the distance between those two genes are very less. So during reproduction, it is easy to get inherited. If the genes are nearby, the chances of inheritance together, it is more. Okay. If the distance between genes, it's more, means the chances of inheritance, it is less. Got it? So in a map, the closer the genes, the chance of inheritance together is more. If the genes are far, there is a less chance of inheritance of those genes together. Okay? So genetic map is created on the idea of linkage. Okay? So the genetic map was created by using the information on polymorphism. Polymorphism or restriction endonuclease recognition site. Okay, restriction endonuclease recognition site. Based on that information, the genetic map was constructed, and some DNA sequences. They found that some DNA sequences which are repetitive, that repeated DNA sequences they called as microsatellite. Some DNA sequences we can see as repetitive units. They are called as microsatellites. Okay, this later we'll discuss in detail about these microsatellites. So here the sequence of chromosome 1 was completed in May 2006. So another challenging task was assigning the genetic and physical map of this genome, and that was generated using information of polymorphism of restriction endonuclease recognition site and some repetitive DNA sequences also, which are known as microsatellite. Okay. So if the genes are near, there is a chance of inheritance. Together they can be inherited. If the genes are far, there is a very less chance of getting inherited together. Got it? Next, features. Let's see the silent features of human genome. So in human genome, around 3,164.7 million base pairs are present. Okay. We said in human genome 3 into 10 raised to 9 base pairs are present. So average gene, it is having around 3000 bases. But the size of these genes, it varies from gene to gene, and the largest known gene of human genome is dystrophin. Dystrophin, that is the largest known gene of human genome. Note down, these are important points, the salient features in this. So many MCQs are there, and salient features of human genome, that is important for board exam also. So what are the important points? Human genome consists of 3 into 10 raised to 9 base pairs, and average gene is having around 3000 bases. Average gene, it is having 3000 bases, but the size of genes, it varies. So based on that, number of genes, number of bases, that also varies. So the largest non-gene of human genome is dystrophin, which is the largest non-gene in human genome, which is the largest gene, dystrophin. This gene is having around 2.4 million bases. So average gene is having 3000 base pairs. But the largest gene, dystrophin, in human genome, that is having 2.4 million bases. So that is the largest one. Okay. And the total number of gene is almost 30,000. So almost 30,000 genes are present in human genome. Okay. So in all people, most of the nucleotides in all the people exactly that will be same. Most of the nucleotides. So 99% will be similar. That shows similarity. Okay. So among these discovered genes, sequencing over. So among these discovered genes, about 50% of genes, their function is unknown. Okay. Still, the 50% of genes function is unknown. It's not proved. And in this, only around 30,000 genes are there. So among that, 50% of the genes function also unknown. Okay. Function, it's not yet known. And among the known ones, only 2% of genes, they are coding for protein. Got it? Only 2% is coding for protein. Okay. So the human genome contains 3 into 10 raised to 9 base pairs, and the average gene. An average gene consists of 3000 bases, but the largest non-human gene in human genome is dystrophin, and that is having around 2.4 million bases. And the total number of genes, it is around 30,000, and among these 30,000 genes, around 50% are having unknown functions. We don't know the function of those genes. And among these, or in human genome, only 2% of the genes, they are coding for protein. Got it? Next, and few more features of human genome project we can discuss. That is, in human genome, very less portion is covered by repeated. Very large portion. A large portion is covered by repeated sequences. Okay, these repeated sequences are covering large portion of human genome, and these repeated sequences, they are DNA sequences, and they are repeated several times. Some repetitive sequences are present in human genome. They are repeated many times, and they covers almost most major portion of human genome. And then sometimes in hundreds or in 1,000 times also they are repeated. So these repeated sequences, these sequences may not have direct coding work. Okay, they are repeated, and these non-coding, they are covering most of the regions of human genome. So they don't have direct coding work, or they are not involved in coding function. Okay, but these repetitive sequence, they gave an idea about chromosome structure, evolution, and dynamics. Okay, So this repetitive units, they are covering majority portion of genome. So they gave idea about, actually they don't have direct involvement in coding, but they gave an idea about the chromosome structure, evolution and dynamics. So among all these genes, that Y chromosome that has least number of genes. Y chromosome is having least number of genes. Note down all these points. Y chromosome is having least number of genes, and chromosome 1. How many genes? Y chromosomes? How many genes? Y chromosomes having 231 genes. But chromosome 1 has more number of genes. Around 2,968 genes are present on chromosome number 1, and Y chromosome is having least number of genes, around 231 genes. So in human genome, scientists, they have identified about 1.4 million locations of SNP or SNPs. What is that? What is SNPs or SNP? Single nucleotide polymorphism. In all these locations, 1.4 million locations of SNPs or SNPs. What is meant by SNP? Single nucleotide polymorphism. So in all these locations, the difference or the polymorphism is just a single nucleotide variation. Okay, they found out 1.4 million locations, which is varying only with single nucleotide. Okay, there is a single nucleotide difference in between all this. So information makes more ideas about chromosomal locations for sequencing, which are these are associated with major diseases. So they got more information through this Human Genome Project, and they were able to identify the genes on particular chromosome, and they found out the number of genes per chromosome, and Y chromosome that is having least number of genes, around 231 genes, and chromosome 1 that has most genes, around 2,968 genes are present on chromosome number 1. Okay, and scientists identified 1.4 million locations with single nucleotide polymorphism or SNPs. They are called a single nucleotide polymorphism. There is only single nucleotide difference that occurs in humans, and this information helped in the process of finding chromosomal locations for disease which are associated with particular disease and sequencing of such things. Okay, that helped in tracing human history also. Got it? Next, applications and future challenges. Where we can apply this Human Genome Project, these techniques and technology or the knowledge which we got from Human Genome Project, where we can apply, and what are the future challenges of this. So one of the great impact of having human genome sequence may well be enabling a radically new approach to biological research. Okay, so in the past, researchers studied one or a few genes at a time. Okay, with whole genome sequence and new high throughput technologies, we can approach questions systematically and on a much broader scale. So they studied one gene or few genes at a time, the sequencing, and from this, the whole genome sequence, and with this hi-fi technologies, we can approach everything in a broader scale. So they can study all the genes in a genome. Okay. So here for an example, what we are saying, all the transcripts in a particular tissue or organ or tumor, or how tens of thousands of genes and proteins work together in interconnected network to orchestrate the chemistry of life. We can study different genes and what are the function of each gene and how it is working, how single gene is working, how so many genes are working in a coordinated manner to perform a function. All this It's very easy to find out with this, or that is the application. There we can apply this because all genes are sequenced and we know on which chromosome which gene is present. So the studies became more easier and it was very easy. Now it became very easy to find out or trace out the disease and the history of disease, everything. Got it? So that is the application. Next is DNA fingerprinting. Okay, DNA fingerprinting. So we are going to learn about DNA fingerprinting. So in DNA fingerprinting, this involves this is an important, very important topic. That's about DNA fingerprinting. So what is DNA fingerprinting actually? DNA fingerprinting, it is also called as DNA typing or DNA profiling or DNA genotyping. All these. Okay, DNA fingerprinting is also called as DNA typing, DNA profiling, genetic fingerprinting, or it is otherwise called as genotyping. So in DNA fingerprinting, what they are doing in DNA fingerprinting? So scientists first they are identifying the difference in DNA sequences. What they will do? Fingerprinting, you know, fingerprint actually what we will say fingerprint is unique. So DNA fingerprinting means like that there is in the DNA profile, it is unique. Okay, so each organism it is having unique DNA profile. So scientists they are first identifying the difference in the DNA sequencing. So we said about some repetitive sequence just before. In the previous topic, we discussed about the repetitive unit or sequences that covers most of the genome. In human genome, most part it is covered by that repetitive sequence. Right, just now we discussed. So they are finding differences in that repetitive DNA sequences. And these small stretch of DNA that repeats many times, sometimes thousands, 10 times and 1,000 times also they may repeat. So first step is separation of this repetitive DNA from the bulk genomic DNA. So this is done by density gradient centrifugation, based on density gradient centrifugation. Okay. So first, what they will do, isolation of genomic DNA, then separation of bulk genomic DNA from the repetitive small stretch of DNA that which is repeating many times. So the separation of this bulk DNA and the repetitive DNA, and that is done by density gradient centrifugation. So during the density gradient centrifugation, this bulk DNA shows one peak and that repeating DNA that's showing another peak. Okay, what is this centrifugation actually? Centrifugation means the process of centrifugation in that the separation of substances based on size that happens. In centrifugation, the separation of substances based on their size. Okay, the centrifugation or the centrifugal force. That force pushes larger particles from center towards the bottom and the smaller ones will be on top. Got it? So the force, the centrifugation force that makes the separation of larger particles and smaller particles. Larger particles will be at the bottom and smaller particles towards upside. Okay, the smaller ones will be towards top. So in the DNA fingerprinting, the repetitive DNA, that repetitive DNA that is separated from the bulk DNA with the help of density gradient centrifugation. So all these all shows different or separate peaks. Okay, so the bulk DNA will be more. Definitely that bulk DNA will be more. So that shows a major peak. Bulk DNA will be more in quantity, so that shows a major peak. And smaller peaks shows satellite DNA or these repeating units. So these satellite DNA, it is classified into different categories. And on what basis it is getting classified? Based on the length of that segment, then number of repetitive units, and based on base composition. Base composition in the sense adenine and thymine rich. If in that sequence adenine and thymine combination is more, that means it is rich in adenine and thymine. And if it is rich in guanine and cytosine combination, GC rich, guanine and cytosine, it is more in that, we will tell GC rich. Okay, so based on base composition, the number of repetitive units, the length of that segment, based on all these things, that microsatellite, the satellite of DNA is classified, and based on all these, the repetitive units are classified into microsatellite and mini satellite. Okay, so based on all these criteria, that satellite or the repetitive units, they are classified into microsatellite and mini satellite. Got it? So important point is that these sequences doesn't code for any protein. These repetitive sequence or repeating sequences or satellites, that doesn't code for any protein, but they are covering majority of human genome. But they are not coding for any protein. But as we said, these forms major portion of human genome. Okay. So fingerprinting involves identifying difference in some specific regions in DNA sequences called as repetitive DNA. So these sequences, repetitive sequences, they are small stretch of DNA. It is repeated many times, and these repetitive DNA are separated from bulk genomic DNA as different peaks during density gradient centrifugation. So the bulk DNA forms major peak, and the satellite DNA that forms the smaller peak. Okay, and based on the base composition, that means it is AT rich or GC rich, and based on length of the segment, then based on number of repetitive units, based on all these things, that satellite DNA or that repetitive units, they are called or they are classified into many categories like microsatellite, mini satellite and all. They are classified into many categories. So these sequences are normally not coding for any protein, but they are forming major part of human genome. Okay. Then this DNA fingerprinting, that involves identifying difference in some specific regions in DNA sequences called repetitive DNA. And these sequences, just now we said, this involves the difference in some specific regions in DNA sequences called repetitive DNA. That repetitive DNA sequence difference is finding out in this. And in these sequences, small stretch of DNA is repeated many times. And this repetitive DNA, they are separated from bulk DNA through density gradients and centrifugation. And this bulk DNA that forms major peak, and other small peaks, they are from the satellite DNA. Then, who proposed this technique? Sir Alex John Jeffrey. He is a British geneticist and known for developing this technique for genetic fingerprinting and DNA profiling. Now we are using worldwide this technique developed by British geneticist Alex John Jeffrey, the technique of genetic fingerprinting and DNA profiling. Alex Jeffrey, he is a British geneticist, and we are using this technique in forensic science and to resolve the paternity issues. Okay, for all these things we are using this technique. Got it? Next. So depending upon the base composition, AT rich or GC rich, then length of segment and number of repetitive units, the satellite DNA is classified into many categories. Why I am repeating all these things means because based on all these, the DNA fingerprinting you can write. You should be clear with this DNA fingerprinting. Then only you can think about the application level of that. And the repetitive units of the satellite DNA is classified into many categories. Which are those categories? Microsatellite and mini satellite. And these sequences, they are not coding for any protein, but they are covering large portion of human genome. So the sequence that shows high degree of polymorphism, and that forms the basis of DNA fingerprinting. So based on the polymorphism of this satellite, DNA fingerprinting is done. So that is the basis of the DNA fingerprinting technology. So since DNA from every tissue from an individual that shows same degree of polymorphism, they become very useful identification tool in forensic application. We know the forensic application for criminal cases and all, we are finding out we are doing this DNA profiling to find out. And the polymorphism, these polymorphism, they are inheritable from parents to children. So DNA fingerprinting it is used for paternity testing also. Got it? Here you can see DNA, this bands will form while checking. So based on those banding pattern, we are doing this forensic application and the paternity testing. Then polymorphism. We have discussed about DNA polymorphism, or that is the basis of genetic mapping of human genome. Okay, and based on that, the DNA fingerprinting is also working. So polymorphism means the difference, or the differences or the changes from one DNA sequence. So how these changes happens? Mainly the polymorphism that appears due to mutations. So these changes start from the cellular level, right? Mutations start from the cellular level. So either in germ cells means sex cells, or in somatic cells. These changes are happening in somatic cells or in germ cells. So suppose in germ cells or in sex chromosomes, these mutations occur, then these type of mutation, it is not at all affecting the reproductive capacity of an individual. Then through reproduction, if nothing is happening to mutation is taking place in sex cells or germ cells. Okay, but that is not at all, that mutation is not at all affecting the reproductive capacity of that organism or that human. Then through reproduction, that mutation or that change which is made into that particular germ cell or sex chromosome, that can be transferred to next generation also, right? So like that simple mutation can be passed to the next generation and next population through reproduction, because it is not affecting the reproductive capacity of that organism. So slowly it is moving to next generation and to next members, right? So that allelic sequence variation, or that difference in the sequence of alleles, it is also called as the DNA polymorphism, and more than one variant at a locus, it is present in human population, or that frequency is more than 0.01%. Okay, so allelic sequence variation it is described as the DNA polymorphism when it is more than one variant, or in allelic level more than one variant is seen, then only it is called as a polymorphism. Okay, at a locus more than one variation, more than one variant in allelic level it is seen, means it is considered as a polymorphism, and in a population that should be seen, or the frequency of that should be more than 0.01%. Then it is considered as a variation or a polymorphism. Got it? So here you can see the blood sample is collected, DNA is extracted, then PCR amplification, agarose gel electrophoresis, and here the banding. Got it? Here the graphical representation. Okay. Then if an inheritable mutation is observed in a population at high frequency, it is referred as DNA polymorphism. So we said that frequency should be more than 0.01%, and the probability of such variation to be observing in non-coding DNA sequence, that would be higher, as mutation in these sequence may not have any immediate effect or impact in individual's reproductive ability. This is what we said, that is not affecting that individual, that mutation which is happening to that gene or to that coding region or that region, that specific DNA region, that is not affecting the reproductive ability of that organism, then it is passing from one generation to next generation through reproduction, right? So these mutations keep on accumulating generation after generation because it is not affecting the reproductive capacity. So that changes, it is passing from this organism through reproduction to next generation. And like that, that mutations are accumulating. So that variability, that forms the polymorphism. So this is forming different type of variations. So there is a variety of different type of polymorphism ranging from single nucleotide change to very large scale changes. So that change or mutations or that polymorphism may be from single nucleotide to very large scale. Okay. Sometimes it may be single nucleotide, sometimes it may be more than that. So like that evolution, for evolution and speciation, this kind of polymorphism are having very important role. Got it? Then DNA fingerprinting technique. So the technique of DNA fingerprinting, it was initially developed by Alex Jeffrey, we told already, and he used a satellite DNA as a probe, and that shows very high degree of polymorphism. And this is called as variable number of tandem repeats. What it is called as variable number of tandem repeats. Got it? So the DNA probe, what do you mean by DNA probe? They are stretches of single-stranded DNA. Single-stranded DNA stretches, and these are used to detect the presence of complementary nucleic acid sequence by hybridization. Okay, while hybridization technique, these are able to find out the complementary strands. So that's how these are working. DNA probes are working. So usually these DNA probes, they are labeled. They are labeled with radioisotopes or biotin or fluorophores. So why it is getting labeled? Because for the detection of these probes. Okay. So in his method, Alex Jeffrey, he used a satellite DNA as a probe, and that shows very high degree of polymorphism. And this is called as variable number of tandem repeats. Okay. And this technique, as used earlier, that involves southern blot hybridization using radio-labeled VNTR, variable number of tandem repeat as a probe. Okay, the southern blotting and all in biotechnological application level, in further chapters you can learn about this. So now how it is working? Here you can see parental chromosome, chromosome number 7, chromosome number 2, chromosome number 16. And you can see this is paternal chromosome and this is maternal. The second one is maternal chromosome. This is paternal, these two, and this is maternal. Down one is maternal from mother and top one is from father. And in chromosome number 2 also, this is from father, the top one is from father and down one is from mother. And same thing in chromosome number 60. This is DNA from in here. This is the control one. Okay. And here in first slot, they loaded the DNA from individual A. This is for checking. Like this only in forensic level, they are checking. Are the paternal issues, how they are confirming the child is from which one. Okay, how they are checking from individual A, they are collecting DNA sample. From individual B, they are collecting DNA sample. And here, this is the DNA sample, the control one. And here you can see the number of short tandem repeats from 11th chromosome. Here it's present. The paternal one is present and the maternal one is also present. And chromosome number 7, paternal one is present here, maternal one is present here. This banding, you can see this banding. And chromosome number 2, the paternal banding is here on top and maternal banding is here. So next, what we are doing, we are going to compare this DNA from individual A and B here. So the first individual, it's the paternal band is missing here, right? But in the B, the paternal banding is here and maternal banding is also present. And in the chromosome number 7, here, it's not matching. It's not matching with the control one. But in the case of individual B, that banding here, it is matching. Matching banding is that banding, same banding pattern you can see in the case of B. And in the chromosome number 2, in that case also, here one band is present here, but the second one is not there. But here in the individual B, you can see both banding. Okay, so finally we can confirm. It shows the percentage of matching level. It is with B. Okay, the DNA from individual B. So like that, we are concluding the parental issues. Okay, so isolation of DNA is done. Then digestion of DNA by restriction endonuclease. Then separation of DNA fragments by electrophoresis. And then transferring of separated DNA fragments to synthetic membranes such as nitrocellulose or nylon. This is the southern blotting. Blotting techniques comes here. And then hybridization using labeled VNTR probes. Okay. Then detection of this hybridized DNA fragments by autoradiography. So these are the steps in DNA fingerprinting. First, we are isolating the DNA. Then digestion of DNA by restriction endonuclease. Then separation of DNA fragments by electrophoresis. Then transferring of separated DNA fragments to synthetic membranes. There comes the blotting technique and hybridization using labeled VNTR probes, and then detection of this hybridized DNA fragments by autoradiography. Okay. Then here VNTR, that means VNTR belongs to VNTR or variable number of tandem repeats. That belongs to the class of satellite DNA. Okay, this is referred as mini satellite. Got it? This is referred as mini satellite, and the small DNA sequences, they are arranged tandemly in many, and the copy number of this will be more. And the copy number from chromosome to chromosome, this copy number varies. In each individual, from chromosome to chromosome, the copy number varies. And this repeats, that shows polymorphism, the degree of polymorphism, that number of repeats, that shows very high degree of polymorphism. The copy number varies and the number of repeats shows very high degree of polymorphism. So VNTR, the VNTR size of VNTR, that varies from 0.1 to 20 kb. Here you can see. This is how the banding will come after checking. Okay, that banding pattern. And after hybridization, the VNTR probe, the autoradiograph, that gives many bands of different sizes. Here you can see many bands of different sizes. Here the top banding, that shows high molecular weight. If weight is more, that will move less distance. If those who are having lower molecular weight, that will move very fast and that will reach down. See, the top banding shows high molecular weight. The size, depending upon the size, banding size, you can see many bands here. And these bands gives a pattern of an individual DNA. Okay, this banding shows the pattern of an individual DNA. And next, the application, DNA fingerprinting applications. So from individual to individual in a population, that this banding pattern of this DNA, it differs. So in identical twins, you can see same type of banding pattern, identical twins, because their genetic constitution also will be identical, right? So the sensitivity of technique that has been increased by use of polymerase chain reaction, PCR reaction. Through that polymerase chain reaction, that application or the sensitivity, that reaction and the application, it has become more sensitive. Okay, so the application that becomes, the sensitivity of this technique has been increased by usage of PCR reaction. Then the DNA from a single cell, it is enough to perform DNA fingerprinting analysis. See, DNA fingerprinting in crime scene and all, we can see suspect 1, suspect 2, suspect 3, a blood sample or hair hair cells. That much is enough. Very less sample is enough to finalize or to get a result. That much less sample is required. First sample collection, then PCR reaction, then electrophoresis, and then banding. So crime scene here for matching with suspect 2, it is here the banding pattern, it's less matching with that. Okay, suspect 1's profile, suspect 2's profile, suspect 3, third suspect's profile. So which one is matching more? Suspect 2. From the crime scene, blood sample is collected and that is trying to match with suspect 1's blood sample, 2 and 3. But when the testing is done, it is matching with suspect 2. So like that, DNA profiling or fingerprinting we can do. Okay, so the applications are in forensic science we can do apply this technology, and in determining the population and genetic diversities we can do this DNA profiling, and many different probes are used to generate DNA fingerprints. Okay, got it? So that's all about DNA fingerprinting. So we have learned all this molecular level applications and the molecular level of inheritance in this chapter. So if you have any doubt, you can ask me. Hope everything is clear. So that's the end of molecular level of inheritance of chapter 6. Thank you.